Yes, good evening to everyone. Welcome back to the week 10 question answer session of the NPTEL course Fundamentals in Protein Chemistry. Okay, today we will talk about protein macromolecule interactions. And here today we'll discuss about protein carbohydrate interactions and protein nucleic acid interactions. Yes. Can you please mute yourself?
ఇంకోటి ఇప్పిరు అన్నారు కదా.
Can you please mute yourself?
సేమ్ ఇదే సేమ్ ఇదే.
ప్రియాంక అత్త స్కూల్ రాగడ పెట్టావు
Yeah, I think the same. Okay. Yeah, so we can begin now. Okay. All right. Let's start with today's class. Okay. Right. So in today's class, we'll talk about protein macromolecular interactions. Okay. And so first starting with protein carbohydrate interactions. So protein carbohydrate interactions are important in our everyday life. For example, this is very important when it comes to immunology. All right. So you have heard about lectins, I'm sure. Lectins are proteins that bind to carbohydrates. Okay. And lectins are important in the complement system of our body. So we will talk about lectins in details in today's class. Then there is a lot of interaction between proteins and carbohydrates when it comes to post-translational modification of proteins. Okay, so in post-translational modifications, there is addition of these carbohydrate moieties onto proteins, and in the previous classes we have seen that your membrane proteins, these are heavily interacting with carbohydrate molecules. Okay, besides that, transport and cell-cell contact, cell-cell communication, cell adhesion, and a lot of different signaling pathways involve protein-carbohydrate interactions. Okay, most interestingly, the blood groups that we know of. There are many different divisions of blood groups. So the most commonly used forms are ABO system and the RH system of blood grouping, right? So in ABO system also, these A, B, all of these different antigens are nothing but interactions of proteins with carbohydrates, different kinds of oligosaccharides. Okay. So now you understand how important protein carbohydrate interactions are. Okay, so this is a very important and very interesting subject. Here we will talk only about the things that are needed for our MCQs. But then this comes under omic studies, which is a very important and very vast subject. So you can read about it. Okay, I really love omic studies and everything. So I would recommend you to read about all of this. There are many different biophysical, biochemical techniques to study all of this. There are high throughput techniques. We will discuss about microarrays today, and we'll talk about a few more other techniques. All right. So let's start. Now, this protein and carbohydrate, how do they basically interact? Proteins and carbohydrates, just like protein-protein interaction, there is a lot of specificity involved sometimes. Okay, there can be stereospecificity. That is, a particular protein is binding to a particular carbohydrate moiety only when there is a particular arrangement of the chemical bonds. So that is stereospecificity. So basically it is binding to one stereoisomer and not to another one. Okay, and there can be high specificity. It can also bind to a particular group, general specificity that can also be there. And then there are a lot of non-covalent interactions. For example, hydrogen bonds, there can be hydrophobic stacking, and there can be these different non-covalent interactions. Okay, so we'll move on. All right, so this is the one about ABO blood group, and this antigen that is attached to RBC, this round thing that you see here, this is actually RBC, and to the RBC glucose is attached in all the three types of antigens. So the blood that contains A antigen is A blood group. Similarly, there is B blood group in which B antigen is attached to RBC. For O blood group, this H antigen is present, and A and B are nothing but modifications of this H antigen. All right, and in the blood serum, the complementary antibody is present. So antigen, if A antigen is there in the blood, then B antibody will be there. All right. So these are the different oligosaccharide and carbohydrate moieties that are attached. So there is glucose, there is galactose, there is N-acetyl glucosamine, and then there is glucose. And N-acetyl galactosamine is there in A antigen. There is galactose in case of B antigen, and H antigen does not have these particular groups. Okay, so these are the different common protein carbohydrate interactions that go on in our body. Okay, there is a table given. Yeah, so there is a table given for your help, right? You can go through all of these. You might already be knowing about all of this, but it's good to just brush up. So lectin carbohydrate interaction, this is important when it comes to the immune response in our body. We'll read about that soon. Then there is glycosylation. I told you about protein post-translational modifications, right? So this is very important when it comes to cell signaling and protein stability as well. Then there is selectin carbohydrate interactions, again important in immune responses. And then heparin and growth factors, this is important for blood coagulation. Lastly, we have ABO blood group antigens. This is important once again for blood compatibility and immune response. Okay, so we'll move on and we'll talk about proteoglycans. So proteoglycans is a group of glycoproteins. So glycoproteins, as you know, these are proteins that contain a carbohydrate moiety attached to them. So proteoglycans is a particular class of glycoproteins found in the extracellular matrix. It is also found in the intracellular matrix, but less abundant. They are made up of glycosaminoglycans, which is the basic unit, and these glycosaminoglycans are covalently attached to the protein core. So if you look at this figure, The one on the left shows you how these proteoglycans are arranged in the extracellular matrix. And if you look at the zoomed in diagram, then you will see that this is actually the protein core, and this the feather-like structure that you're seeing, that is the protein core, and this long structure is actually hyaluronic acid, and then to that you can see that these glycosaminoglycans are attached. These glycosaminoglycans can be chondroitin sulfate, there can be keratin sulfate, and all of these different molecules can be there. Okay, so we'll talk about them more. Okay, so this is the structure of the proteoglycan, and this you realize is the protein core. This is a residue to which this is actually a serine residue, and to this serine residue 4 monosaccharides are attached. So this is xylose, then there is galactose, and this is glucosamine, and this is your glycosaminoglycan, which is the GAG, and this is made up of one is an acetylated sugar and there is a uronic acid. So it is very important that you remember the structures. For the structures, please refer to either Voigt and Voigt, you can refer to Schreier, you can refer to your Leninger. All the links are provided, okay, in the reference slide. So please go through them. Remember the structures of all of these, all right? Okay, so now we have a question. So the question asks you, which among the following is or are true about proteoglycans? You try to answer this, you can write down in the chat box, okay? So which among the following is true about proteoglycans? The first option says they are present in both extracellular and intracellular matrix. Second option says only one sulfated glycosaminoglycan chain is covalently connected to the membrane protein. Third option says glycosaminoglycan is connected to the serine residue through a tetrasaccharide bridge. Heparan sulfate and chondroitin sulfate are types of proteoglycan. So which of these do you think is correct? C. Okay, right. Let's look at the right answer. I think this is there in your this year's questions also, if I'm not wrong. Okay. Yes, you guys are correct. So one or more sulfated glycosaminoglycan chain can be covalently connected to the membrane protein, right? So that means your 2. Only one sulfated glycosaminoglycan chain is covalently connected. That is not true. We can see that more than one glycosaminoglycan chain can be connected to the membrane proteins. Okay. Right. Now we learn more about proteoglycans. Okay. So we'll look at examples. There are proteoglycans known as syndicans and glypicans. So these are 2 families of the heparan sulfate proteoglycans. So syndicans are majorly transmembrane proteins. So if you look at the figure, you will see that they are majorly transmembrane protein with a single membrane spanning domain. Whereas if you talk about glypicans, these are actually proteins that are attached to the glycoside phosphatidyl inositol, which is GPI. So they are basically anchored to GPI, and meaning that they are attached to the cell membrane via lipid tails. Okay. Now you need to look at the structures. So this is how they are, and these are the different components that make up these different proteoglycans. All right. So we have one more question here. Syndicans and glypicans are 2 major families of membrane heparan sulfate proteoglycans. Now you have V, W, X and Y. 4 different blanks. Dash are held by dash interactions between a sequence of dash amino acid residues and the plasma membrane lipids. We know about lipids, right? One particular type is interacting with the membrane lipids However, dash are covalently held by membrane lipids. One is held by your interaction with the protein. So, yeah, interaction between a sequence of dash amino acid residues and the plasma membrane, and the other one is held covalently attached to lipids. So, which one do you think? Okay, we have two answers. Let us see which is the right one. Yes. So, answer option number A is correct because syndicants are held by hydrophobic interactions between a sequence of non-polar amino acid residues and the plasma membrane lipid. So, we talked about syndicants being this transmembrane protein, right? That is why syndicants are basically being transmembrane. There will be non-polar residues, and therefore these non-polar residues and the plasma membrane lipids hold these syndicants together by hydrophobic interactions. And glycans are held by membrane lipids through covalent interactions. So you can read more about these, and that will help you understand them better. Now we have glycosphingolipids. We'll talk about glycosphingolipids in the upcoming slides. Now, glyco and sphingo and lipid. For these oligosaccharides, you will have to break up the name because mostly the structure and everything you will get to know from the name. Alright. So, glycosphingolipids are plasma membrane components in which the dash head groups are dash. D is hydrophilic, and your second blank is oligosaccharides. Alright, let's see. Yes, that is very correct. So, sphingosine unit is this long chain with this alcoholic group, right? With this OH group, hydroxyl group. This is the alcohol part. And then you have the sugar unit, and you have this fatty acid unit, which is here, right? This is what the glycosphingolipid is. Glyco comes from the sugar unit, sphingo comes from this alcohol part, and lipids come from this fatty acid portion, right? And which is the head and which is the tail? That is, the head is always hydrophilic and the tail is made up of oligosaccharides. Okay, so these are different examples of sphingolipid. So see, these are phosphosphingolipids, and here you see that this phosphate group is here, and this is the long fatty acid tail, right? This is the fatty acid, and here is the tail again. These are the different examples. Now these are complex sphingolipids. This is your sphingomyelin, where you have this fatty acid moiety, and this is the long chain, and here is your phosphocholine group. So this is the hydrophobic tail, and that is the hydrophilic head. Okay, right. Now we'll go to the next question. Okay, the next part is actually lectins. So lectins, you know, these are proteins which have affinity for carbohydrate moieties. So lectins are called carbohydrate binding proteins, and these are very important. They are found in almost all organisms. Lectins are important even in the complement system of our body to identify these antigens, pathogens that enter into our body. Okay, so lectins are very, very specific. They specifically bind to carbohydrates or sugars and participate in cell recognition, cell adhesion, signaling, and immune responses. They are very, very stereospecific and are important in the complement system of our body. So here is a figure for the different families of lectins. Examples are given. You can see R type, and then there is L type, and under L type comes your concavalin A. There is P type, C type, and you will see what is the basic difference between all of these different types is basically the binding specificity and the structure. Okay, that is the major difference. You can read about all of these different types. It is there in, I think, the videos which ma'am taught you. So you might already know. Right? So these you must remember. Here I have listed out everything, all the different examples of the different classes of lectins, their binding specificity, and their functions, major functions. Now we have a question. You can go through this table. That will help you to remember everything. Okay. So we have a question, and we will first go through this table because that will help you to answer this question better. Okay. So coming to P type. Under P type comes concavalent A, and the P-type lectins have binding specificity towards N-acetyl galactosamine, galactose, and mannose. They are mainly involved in cell agglutination, immune response, cell adhesion, growth factor signaling. C-type, under C-type comes the selectins and mannose binding lectin. Right. So, mannose binding protein is also very important in biotechnology for purification of your maybe mannose containing components. Okay. So, immune response, and we also have cell adhesion. I type is important for cellular signaling, apoptosis regulation, neuronal signaling and development. A type is important for immune modulation, cell agglutination, immune response and antimicrobial activity. Similarly, L type and F type you can read about. Alright. So, we will go to this question now. So, X type lectins are essential for cell-cell adhesion. Y type lectins are essential for development and maintenance of the nervous system. Z-type lectins are essential for their role in the generation of functional lysosomes. Identify the different types of lectins. X is C, Y you are saying is I, and Z you are saying is P-type. Z you are saying is P-type. Okay. We don't have that as an option. Okay. We'll see. Right? So, the right option given is D. Actually, none is correct. Is that so? Okay, then you can write to the NPTEL team and confirm, because I remember lectins are there in ma'am's videos, and you can also confirm from all the resources that I've given you. Okay, so if you think this question, the answer is not correct, then please write to the NPTEL team and they can help you sort this out. Okay. Right. So we will go to something interesting, and this is oligosaccharide microarray. Okay. So in oligosaccharide microarray, what is happening? These oligosaccharide moieties are taken. Oligosaccharide moieties are immobilized on a plate. Alright. And this immobilization can be through covalent cross-linking. It can also be through non-covalent interactions, and they are actually immobilized. Then what happens? Then the proteins are added which are binding, which have specificity for these different oligosaccharides. And then after that, immunofluorescence detection is performed. The proteins can be labeled with fluorescent. Okay, so the proteins are added, and then after that, you can have maybe antibodies that are fluorescently labeled, and they will detect those proteins, right? There can also be mass spectrometric detection to get the sequence of the proteins that are binding, and then there can be SPR imaging. From there you can actually know about the different binding interactions and the KD values as well, right? So the specificity and the binding affinity of the glycan binding proteins, lectins and antibodies are studied for immobilized oligosaccharides using high throughput oligosaccharide microarray. So this is a high throughput technique. High throughput means at the same time you can get information on many different proteins and many different protein oligosaccharide pairs at the same time. So that is why it is high throughput. So the microarray based technology has been widely employed for rapid analysis of the glycan binding properties of lectins and antibodies, the quantitative measurements of glycan protein interactions, detection of cells and pathogens, identification of disease related anti-glycan antibodies for diagnosis and fast assessment of the substrate specificities of glycosyl transferases. Glycosyl transferases are enzymes that interact with the oligosaccharide group and they transfer this to different molecular entities, right? So yes, this is what is done using oligosaccharide microarray. Right. Now we have a question on oligosaccharide microarray. Okay, try to answer this question. Oligosaccharide microarrays are used to determine, the first option is specificity and affinity of the carbohydrate binding by lectins. The second option is the amount of carbohydrate bound to the lipid membrane. Okay. The third option is the affinity of a particular amino acid residues in the proteoglycans and the amount of glycation in the protein. Okay, the right option would be A, because the rest of it, actually the amount of carbohydrate bound and all of this, it is not possible to detect directly from oligosaccharide microarray. And okay, so from this oligosaccharide microarray you can know about the specificity and affinity of the different carbohydrate binding proteins. Okay, so yes, A is the right option. Right, now we come to something very interesting, which is a test. Okay, this is a diagnostic test that tells you about the amount of sugar present in your blood, and this is an indication of diabetes mellitus, whether you are diabetic, whether you are pre-diabetic, and whether you are not. Okay, so this is HbA1c. From Hb you can understand that this has something to do with hemoglobin, right? So an HbA1c test is also known as a glycated hemoglobin test or A1c test. This is used to measure the blood sugar levels in the body, which aids in diagnosing and managing diabetes mellitus. So the HbA1c blood test value, denoted in percentage, that you can see, okay, indicates the average blood sugar level for the past 2-3 months. Now, HbA1c test normal range includes values between 4 and 5.7%. Less than 6.5% indicates pre-diabetic, and HbA1c value of 6.5% and more indicates hyperglycemia, which is diabetes mellitus. So there is too much of glucose in your blood. Okay, so what happens in this test? So see, this is hemoglobin, and to this N terminus of hemoglobin beta chain, this glucose molecule gets joined. So Hb, one second, why 2 to 3 months? Why not 120 days? That is 4 months. Yes, it is technically 120 days because that is the lifetime of hemoglobin, right? So yeah, technically it is 4 months, but then 2 to 3 months is taken for safety, because you don't know whether the hemoglobin molecule was just synthesized or it was already there in the blood for quite a long time, right? That is why. So hemoglobin A1c is metabolically produced by the reaction of glucose with the N-terminal valine residue of each beta chain of hemoglobin and the subsequent formation of a stable ketoamine. So you know about ketoamine, you know about ketosis, right? Yes, life of RBC. 120 days is the life of RBC, and that is why. Yeah, okay. So a stable ketoamine is formed. So what happens in HbA1c immunoassay? So the pre-treatment and hemoglobin measurement. So whole blood cells are lysed. Problem when repeat. Yes, what is it? Beta chain in RBC. Retest. I am not understanding. Oh, Acha, you're saying that? Okay. Retest is also advised for this assay, and that then there is a problem. Please discuss previous slide. Okay, sure. So see, what happens here is that we will talk about the principle soon. So here what is happening, this is just telling you that there is a glycation event happening. So in glycation, to know treatment is working or not. Yes, to know whether the treatment is working. Okay. So you're saying that it is repeated to know whether it is working or not. That is why for safety it is said that 2 to 3 months instead of 4 months, which is the actual lifetime of RBC, right? Okay. Got it. Okay. Right. So see what is happening in this HbA1c immunoassay. Here what is happening? This glucose is interacting with the N-terminal valine residue of the beta chains in hemoglobin. This is a non-enzymatic reaction, and with this valine residue, why are you not teaching? What is wrong? Is there anything you want to say?
Hello,
Yeah.
ma'am. Beta chain and alpha chain in this cluster. What that denotes?
I'm not able to hear you. Can you please repeat?
मैम बीटा चेन और अल्फा चेन इस क्लस्टर में जो एन टर्मिनस ऑफ हेमोग्लोबिन में बीटा चेन दिखाया गया है,
हाँ,
वो क्या बताया जा रहा है?
देखो नीचे लिखा है. We will read the in the next slide. We have the entire principle of the test.
Okay.
Right. Basically, what is happening? This glucose is actually getting attached to the beta chain of hemoglobin through a valine residue. Okay, that is being shown here. That your hemoglobin is getting attached to glucose residue. That is what is happening in this test. And then after the entire lifetime of this hemoglobin, we will again measure it. So everything is there in the next slide. We'll see that.
Okay.
Okay, we'll see this now. Okay, so what happens is basically when in the beginning of the test, they will take your blood sample, and then there is hemolysis, and hemoglobin is released from the RBC, right? And then they will measure the amount of hemoglobin that is there. So absorbance of the hemoglobin is measured at 570 nanometer after addition of a particular reagent. So you can read about this reagent if you're interested. Then what happens? It is basically proportional to the total hemoglobin concentration in the sample. So that means this is a normalization. They are checking how much hemoglobin is there. After that, they are adding a particular reagent. This is referred to as R2. Okay, you can check again what is the content of this R2. Then what is happening? There is fructosylated dipeptides from the N-terminal hemoglobin beta chain are released by proteases. So hydrogen peroxide is produced after oxidative cleavage of the fructosylated dipeptides by a particular enzyme. H2O2 is determined colorimetrically by reaction with a particular chromogen at a particular your different wavelength. So the absorbance increase is proportional to this HbA1c concentration. So what is happening here? This is the entire principle behind this test. So what is happening? This is initially hemoglobin. So you can see that hemoglobin is having this NH2 group, and this is the one that is there in valine. Now what happens? You are adding glucose molecule. So what happens? This gets attached and the Skipp's base is forming. You know that Skipp's base is going to give you color, right? And that is why there is colorimetric detection. So H2O2 is produced, a different enzyme is used. So if you're interested, you can read about all of these different components, exactly what is happening. But the basic principle is that in this test, they are checking the concentration. They're basically checking this hemoglobin after a few months. That is after, let's say, 3 months. They are checking whether this hemoglobin is still in your blood or not. This hemoglobin attached to glucose, right? And then from there they are determining whether you are diabetic or pre-diabetic or what is the situation in the body. That is what the test is all about. If you're interested, you can read more about that. Okay, so we come to the question. Please try to answer this question. Okay, so you're saying that it is D. So the first option is HbA1c test is a method to detect a modified form of hemoglobin. Which among the following statements is or are correct about HbA1c and or modified form of hemoglobin? So this is used to measure the average plasma glucose concentration over prolonged periods. It is a form of hemoglobin that is chemically linked to sugar. It is formed through an enzymatic pathway. So HbA1c test is used to measure the amount of blood sugar, glucose attached to hemoglobin. This is a modified form of hemoglobin that is actually called glycated hemoglobin, which is formed through a non-enzymatic glycation pathway caused by interaction of glucose and with hemoglobin. So they have asked you which one is false, and therefore only 3 is false. Alright, so we'll move on to the next question. So interaction of proteins with nucleic acid. So with this we come to the end of questions on protein and carbohydrate interactions. You can read about more of these because there is a lot to read about this. For our questions, only this much was sufficient. So I have included this in the PPT. Now we move on to protein nucleic acid interactions. Right. So this is a very general question, and they are asking you the interactions of proteins with nucleic acid are essential for which of the following biological processes? DNA replication and repair, RNA processing and translation, gene expression, regulation of chromatic structure. So yes, all of you are correct. All the different processes are important for protein DNA interactions are basically important for all of these different processes. So yes, D is the right option. Now we will read about the different motifs or different domains in proteins that interact with RNA. Okay, so there are different domains in proteins that interact with RNA. This is one of them. This is called the dead box domain. You can read about all of these different domains. So please refer to the books that I have mentioned. There are different websites where you can read about all of these. So basically, dead box domain is a characteristic feature of a family of proteins known as the dead box RNA helicases. So there is this helicase code, and it is involved in RNA binding, RNA processing, unbinding of RNA, and regulation. Alright, so dead, this term dead comes from the presence of a conserved amino acid motif, which is aspartate, glutamate, alanine, and your aspartate again. That is giving it the name dead. So dead box proteins are typically, they are made up of two Reiki-like domains that facilitate their helicase activity. Okay, so you can see that there is Reiki domain 1 and Reiki domain 2 that help in helicase activity by using ATP and Dead box helicases utilize ATP hydrolysis to change their conformation and promote the unwinding of RNA. So RNA unwinding and change in conformation is achieved. Next we have the K-homology domain. In K-homology domain you can see that there are both alpha helices and beta sheets. There is a very characteristic fold, three-dimensional characteristic fold that is achieved, and this basically beta alpha alpha beta is the domain. Alright, so there is type 1 and type 2 K-homology domains. Type 1 is on the N-terminal side, type 2 is on the C-terminal, and you can see that this is very important for the regulation of RNA splicing, translation and RNA stability. So K-homology domains are made up of 70 to 90 amino acids and they adopt a characteristic three-dimensional structure. It contains two conserved sequence motifs often referred to as K-homology 1 and K-homology 2, which are important for RNA binding. So these RNA binding proteins, a very interesting thing about RNA binding proteins is that they are not very sequence specific all the time. Okay, but when it comes to DNA binding proteins, they are mostly always sequence specific. So that means they are going to bind to DNA of a specific sequence only, generally. But then RNA binding domains, they are not very sequence specific and they can bind to RNA of any sequence. Okay, that is very common in RNA binding proteins. So these proteins bind to RNA without much sequence specificity. Okay, now we have another domain which is S1 and S2. This you can see in the SARS-CoV-2 coronavirus, and these S1 and S2 domains are again important in RNA binding. So they are found in the various RNA binding proteins, particularly those involved in RNA metabolism, splicing, translation and degradation. So S1 and S2 bind to single-stranded RNA and using their characteristic fold of beta barrel structure. So they actually have a characteristic fold which is a beta barrel like shape, and S2 also mediates protein-protein interactions. Okay, we'll try to answer a question. This is the image. This is the structure of a particular nucleic acid binding domain. So they are asking you what kind of domain it is. So you can see that there are repeats. And if you remember these different domains that we studied about, then you will be able to answer this question. There are two repeats. If you remember K-homology domain, it is very small. There is only this beta, alpha, alpha, beta. So this would not be K-homology. Then which one do you think it is? Okay, I'm getting A and C as answers. Let us go back to these figures and see. Right? If you remember this is the RNA binding domain and these are other domains. Right? So you can look at the topology diagram for these also, S1 and S2. That will help you out. Coming to the dead box domain. So dead box domain, you can see that there are so many different components and it is huge. Right? Coming to K-homology domain, you can see that this is how K-homology domain looks like. There are two beta sheets and there are alpha helices. Okay. The right answer for this is dead box domain. Now we go to a technique, a biophysical technique required for studying protein DNA interactions. So this is called footprinting. Now footprinting can be done for DNA, it can be done for RNA. So what happens in footprinting is that, what is a footprint? This is basically, let's say there is a transcription factor. Okay, this transcription factor, let's say this is the DNA and this transcription factor is binding to this DNA like this. Now what we apply is, we apply DNases. So DNase, you know, it is going to come and cleave DNA, right? But then it cannot access the DNA that is bound by this transcription factor. It is not able to cleave the DNA that is bound by this transcription factor. So DNase is going to make tiny pieces of DNA, and when you run all of that on a gel, you will see that there are different pieces, but then the region of the DNA held strongly by this protein is basically missing. So that is called the DNA footprint. And when you take that particular region, so this region will be relatively more intact, okay, because this portion, the rest of the portion will be digested totally, but then this portion will be relatively more intact. And so when you go for sequencing, then you see that you will get the basically sequence with which this transcription factor has specificity. So here you can see in this figure that there is a labeled DNA, and a part of this is taken where this particular DNA binding protein binds, and the rest of it is taken where no protein is bound. This is naked DNA. Then what happens? You go for limited DNase 1 digestion. So after that, what happens? So limited means that partial digestion is happening. Complete digestion is not happening. So you're keeping it for a prolonged duration. Then what happens? You run it on a gel, and for the DNA that is not bound by any protein, you can see small fragments for the entire length of the DNA. But then for the region where you are having this footprint, then you can see that the rest of it is forming tiny fragments, but the footprint will actually be relatively bigger. So now what we do is we select that footprint and we go for sequencing. So this is a very important method for studying protein DNA interactions. So this is a question on that. Try to solve it, which is not true about RNase footprinting assay. So the figure we saw was about DNA footprinting. Here they are asking you about RNase footprinting. So you can understand that there is an RNA binding protein which is binding to RNA, and we are cleaving with RNase. So the rest of the RNA is cleaved and only the footprint remains relatively intact. So then we will go for sequencing and get the specificity of this particular RNA binding protein. So proteins that bind to an RNA sequence can protect that region of the RNA from the RNase digestion, which you know is true. The region that is not protected is known as the footprint. This makes it possible to locate the protein binding site on a particular RNA molecule. This method uses RNase to cut the end labeled RNase with and without bound protein, followed by gel electrophoresis to detect the resulting cleavage pattern, which is not true. Think, which is not true. They're not asking which is true. All of you are wrong. What is wrong with you guys? You did not understand this part. Yes, finally I'm getting right answers. Yes, correct. So it is B, because the region that is protected is called the footprint. Right, the rest of the region is chopped off. So it is not footprint, it is getting chopped off, it will be lost in the gel. Only the region where DNA was bound. Okay, one more very important thing is that now this protein DNA interaction is not that strong, right? So to make sure that the protein is basically bound to the DNA, or RNA binding protein is bound to this RNA, you first need to cross-link this. So cross-link means that there is covalent interaction between the protein and the RNA first. Then after that you will have to decross-link after this DNA's digestion. So when you decross-link, you will get only the DNA, which is actually the footprint. The rest of it will be so tiny that it will be very difficult to see them on the gel. But then the footprint will be relatively bigger and you can go for sequencing. Alright. So that is one important step. Cross-linking and decross-linking come in. Okay. Now we will talk about replication protein A. So replication protein A, in the previous slide it was shown that at the end level DNA is partially digested by DNase 1. Yes, that is true. It is partially digested. Why is it called partially digested? That is because it is not completely digested, because you are having this DNA binding protein bound to the DNA. That is why complete digestion by DNase 1 is not possible. Right? That is why it is partially digested. But option D has RNAs in place of DNAs. Yes. So footprinting can be done for DNA and RNA binding proteins. Right. So when you're using RNA and a protein that binds to RNA, you will go for RNase A. RNase A chops off single stranded RNA. Correct. When you are doing the same experiment with DNA and DNA binding protein, you go for DNase. Got it? Okay, great. So we come to a protein which is a single stranded binding protein. This is useful when it comes to DNA replication because this is a single stranded binding protein. So it is going to, after this DNA gets unbound, right? That is your replication fork is forming, and then the single stranded binding protein comes and binds to the single stranded DNA. Correct? So that is what we can see here in the figure on the left. So this replication protein A is coming and binding to the single stranded DNA during DNA replication to facilitate its single stranded form. Another function of this protein, replication protein A, so RPA, another function of this is DNA repair. So that is homologous recombination and non-homologous end joining. In both of these processes, RPA is involved. So the figure is given and you can read about it. Now, its key role is basically stabilizing single-stranded DNA, which is seen in replication, repair and recombination processes. Alright. So which of the following is not a function of replication protein A? Try to answer this. It prevents single-stranded DNA from binding back on itself or form a secondary structure, which is not its function, remember. So it helps to protect the single-stranded DNA from being attacked by endonucleases. It also helps to bind DNA during nucleotide excision repair process. It regulates the transcription of genetic information from DNA to messenger RNA. Okay, yes, that is right. So regulation of genetic information from DNA to RNA is not its work. It is only involved in repair and replication, right? It is not involved in transcription. Right. We'll go to the next one, which is chromatin immunoprecipitation. See, I told you about these omics studies, omics techniques. Under omics comes genomics, transcriptomics, lipidomics, and then metabolomics, and then there are so many different omics, okay, studies. So under omics studies, all of these different techniques come. Chromatin immunoprecipitation is very important, very commonly used. So in this technique, what happens? Basically, there is a cross-linking between your protein and DNA. So in chromatin, what is there? In chromatin, there is DNA and there are proteins, right? Histone proteins and other proteins that bind. So there is cross-linking. See, we know that DNA-protein interaction is strong, but then when you go through so many different steps, then what will happen? It will definitely not be binding to each other anymore. So we need the binding to be consistent, and for that, covalent cross-linking is very important. So you treat it and then covalent cross-linking happens. Then after that, you break open the cell and you get basically lyse the cell, and then you are getting the chromatin in which protein and DNA are binding together. Then what happens? Then the antibody comes and binds. So there are specific antibody against the proteins that you want to study, And then what happens? This is basically immunoprecipitation, where the antibody is used, and this antibody is, let's say, labeled, and then what happens? You are washing it to remove the non-specific interactions, and then after that you are reversing the cross-link. That means you are removing this protein DNA interactions, right? So now these are separated. Then what happens? Then only the DNA footprint remains, and you separate that DNA out. So there is purification, there is quantitative PCR. So in quantitative PCR, what is happening? You are amplifying it and you are converting it into what? Basically, quantitative PCR, you are basically amplifying it, and from here you can actually study both the DNA and the protein. Okay. So this is very useful, and from chromatin immunoprecipitation you can also take the you can take the DNA for, let's say, sequencing, and you can know about its sequence, and you can isolate it, and you can study both the protein and the DNA interactions in vitro as well. So this is actually it's a very useful assay. Okay, so what happens in chromatin immunoprecipitation is given here. All the steps are given. So you know that firstly there is cross-linking, then there is chromatin isolation. So the cross-linked chromatin is lysed, the cell is lysed and it is isolated. Then what happens? Immunoprecipitation. In immunoprecipitation, the antibody specific to our protein of interest is binding to the protein which is bound to the DNA of interest. Then what happens? Then there is washing, as we talked about, reverse cross-linking, and DNA purification using techniques like PCR, quantitative PCR, or chip sequencing, which is basically DNA sequencing after chromatin immunoprecipitation for the footprint DNA that is carried out, and then you can study both the protein and the DNA of interest. Okay. Now there are different variations of this chromatin immunoprecipitation. Okay, so these are very important. You get these as match the following. You get these as MCQs also. Chip is very important. Chip on chip is also very important. So in chip on chip, what happens? Genome-wide analysis of the protein binding sites using microarray analysis of the purified DNA fragments. So this first chip is for chromatin immunoprecipitation, and the second chip is for microarray chip. Okay, so here you can basically study about a particular protein that binds to everywhere in your genome. So that is genome-wide analysis of protein binding sites using microarray analysis of a purified DNA fragments. So there can be different antibodies binding to different proteins, and you can study all of them at the same time. You can study the different DNA binding sites of the same protein. Okay, it is very diverse. Then we have chip sequencing. Chip sequencing, you know that this footprint DNA you take and then you go for sequencing. So genome-wide analysis of the protein binding sites using deep sequencing of the purified DNA fragments. You have native chip, in which this omits the cross-linking step and uses micrococcal nuclease digestion. This is MNase. MNase is a useful enzyme. So this digests the DNA at the histone linkers to examine the DNA target of the histone modifying enzymes. So there are different enzymes which are involved. This is all about epigenetics. So in epigenetics, what happens? There are enzymes that are involved in adding some kind of marks. Okay, some modifications are done to the DNA. So let's say that methylation is done, or let's say phosphorylation is done. Okay, and these different marks tell the genes when to express. Often methylation at a particular DNA, and then methylation in chromatin, the histone proteins, these are very important epigenetic markers. So basically in native chip, what happens? We are not using cross-linking But then we are using the native DNA protein interactions. We are using MNAs, which is micrococcal nuclease, to digest the DNA at the histone linkers. So linker means you know about histone proteins. You know that histone proteins are made up of 5 different types. There is histone 1, 2, 3, 4, and in between 2 histone octamers, there is the region where you get this linkers, right, to examine the target of the histone modifying proteins. And then we have chief exo, that is addition of an exonuclease digestion step. DNase 1 is endonuclease, but then chief exo uses exonuclease digestion step to obtain increased resolution of the protein binding sites. So all the extra sequences are removed and up to single base pair resolution can be achieved. Right. The next one is pull down assay. So in pull down assay, we have a particular tag which binds to a particular protein, and the other proteins that interact with this protein of interest also come out when we purify this particular protein of interest from the whole cell lysate, and this is called pull down assay. Okay. So for example, here they have used a GST tag, and this is basically binding to a particular bait protein, and this glutathione affinity resin is used, and the bait protein is binding to this GST tag, and the all the other proteins that bind to the bait protein are also getting purified. So that is a pull down assay. Okay, now we have a question. Try to solve this. Which of the following methods is or are used for the detection of protein nucleic acid interactions? The first one is pull down assay. The second one is electrophoretic mobility shift assay. We learnt about EMSA in the previous slides, right? And we have tRNA footprinting assay, and we have chromatin immunoprecipitation. D again. So D is 1, 2 and 4, right? Yes, that is correct, because you know that tRNA is DNA footprinting assay is used instead of this tRNA footprinting assay. Okay, because tRNA is binding to amino acids and it is also binding to your ribosome, and it is recognizing the codons in DNA, but then this is not used as such. Okay, so next is a very general question. Of course, you know this. Yeah, it is used in translation. Oh, sorry, not, yeah, right. It is useful in translation, correct. So it is basically recognizing the codons in your mRNA. Okay, so we have the next question. The histones have a dash content of dash as well as dash charged amino acids. So histones are binding to DNA. DNA is negatively charged. So what amino acid should be there in histone? I remember studying this in school. B. B is high concentration of lysine and arginine. Yes, very correct. Next is lacoperon. I'm sure you remember lacoperon. So what happens in lacoperon? You can tell me. Basically in lacoperon, what happens is it is a set of genes that are present together, and yes, repression is happening, repression of the genes, and there is a lac repressor which is made by this particular gene, LAC-I gene, and the lac repressor is going and binding to the operator region. Operator region is very close to the promoter, and so RNA polymerase is not able to come and bind to the promoter region. Therefore, it is not able to express the downstream genes. But then when there is an inducer, it is coming and binding to the lack operon, it is lack repressor, and the repressor's conformational change is preventing it from going and binding to the operator. So then your RNA polymerase is coming and sitting at the promoter region, and it is allowing the formation of all of these different genes. Okay. Now this is a question. You can answer this. Question on protein macromolecule interaction. A DNA binding tetrameric protein with 4 identical subunits operates by a helix turn helix motif, which is a very common DNA binding motif. We will read about this very soon, which inhibits the expression of genes coding for proteins involved in the metabolism of disaccharide given in bacteria by reducing the transcription of proteins. What is the name of the DNA binding protein? And how does the bound repressor reduce the transcription of the specific proteins? B. It is a lack repressor protein. It functions only by obstructing the RNA polymerase binding site. Correct. Okay. So here they are saying that there is basically prompting DNA looping obstruction, but I would say that this is seen in tryptophan operon. Alright, so in trip operon we know that there are DNA looping, and that is how transcription is prevented, right? So in my opinion, the right answer should be B. If this is lack operon, then it should be B. Right option should be B. Okay. Because they are already talking about a disaccharide. So you can mail them and you can confirm with them, because DNA looping mechanism comes when we talk about tryptophan operon. There is this attenuation site and everything. Okay. So you can basically ask them. Okay. Now we move on to the DNA binding protein motifs. So there are different motifs of DNA binding proteins. This is the last question for the day. Okay, so there are very significant motifs which are seen in transcription factors. These are seen in DNA binding proteins. Alright, so one of the very common ones is leucine zipper. So in leucine zipper, what is there? There is basically an N-terminal hydrophilic DNA binding domain for transcriptional activation of the targeted genes. And there is a C-terminal B-zip hydrophobic domain. So with leucine at every 7th position stretching on a length of 8 helical turns, tightly held together by alpha helices of 30 amino acids. This segment is also called a B-zip or basic leucine zipper, present in dimer form. Okay. Now we will see the next motif. So if you have understood, this is how a B-zip looks like. The C-terminal portion is this, and this is the N-terminal portion, which is actually binding to DNA. Okay. Now we have another motif, which is the zinc finger motif. So zinc finger motif is involved in DNA binding proteins, and here what happens, this can be a C coordinating with zinc. So 4 cysteines coordinating with zinc, or 2 cysteines and 2 histidines coordinating with zinc. In different cases, there can be even 3 cysteines and 1 histidines. Okay, that is also there, but very rare. And we see that this is actually coordinating with zinc, and the rest of the thing is present in the form of a alpha helix and beta sheet. So this is how zinc finger motifs look like. And okay, so now this is a question. Helix turn helix, I am sure you remember. There is a helix, there is a small turn of 3 amino acids or 2 amino acids, and then there is another helix. So that is a helix turn helix. So if you look at these figures, you will be able to see. Now you tell me which is a leucine zipper, which is a leucine and helix loop, helix leucine zipper. There is a zinc finger motif and there is also helix turn helix motif. Okay, so we have got answers. Great. Okay, let's see. Yes, you're correct. B is the right answer. So these are the different books and different websites that you can go through. And thank you for, yeah, sure. If you have any question, you can ask. Ajay, do you have any question? Glycosylated HP helpful in pregnant too. Okay. Yeah, you can read about that if you're interested. You can always read about that. Okay. If you don't have any other questions, we can end the session today. Thank you so much for attending the class. Thank you for your time. Thank you for your patience. Thank you so much. Thank you. Let's end the session then. Thank you so much. Okay, someone is joining now, but then our session has ended. So we'll meet again next week and we will meet on Monday. Okay, because Puja starts, Durga Puja starts. So we will not be meeting on Tuesday again. We'll be meeting on Monday. Okay. Okay. All the best for your work. Bye-bye.